Welcome to Ash Jottings. Now let's see the important features of Watson and Crick's double helical model of DNA. So far we actually know that based on X-ray crystallographic studies, Watson and Crick suggested that in a DNA molecule, the helix is formed of two polynucleotide chains arranged anti-parallel or in opposite directions. So you can see that it is a two polynucleotides which are arranged in a way. This is one, this is two, right? Now they are anti-parallel 5 prime to 3 prime and its opposite 5 prime. Sorry, I haven't used my ink. Okay. So it is 5 prime to 3 prime direction, and other one is the 3 prime to 5 prime direction. It means that the 3 dash end of one chain lies besides the 5 dash end of the other chain, right? So in such a structure, the phosphate groups of nucleotides in each polynucleotide strand lie on the opposite of the deoxyribose, and the nitrogenous base are inward. As I mentioned is previously, the backbone structure, that is a phosphate, and the sugar is a part which will be present here, which is hydrophilic, and this is the nitrogenous part, which is hydrophobic, which will be present inside. The two nitrogen bonds are nitrogen bases of the two strands, you can see, are linked together by what is called as hydrogen bonding. So they're formed between the two bases between this polynucleotide. So the unique feature how they are pairing also we have seen, that is adenine will pair with two hydrogen bonding with the thymine, and guanine will have three hydrogen bonding with cytosine. Okay, now due to this type of base pairing, the two strands are they are complementary. They are complementary, right? Because of this type of to each other. That means if a chain has a region with a sequence of nitrogen bases, thymine, cytosine, adenine, so you can just consider an example like thymine, cytosine, adenine, cytosine, and guanine. Okay, so you have, let me write it down, that is more better. You have the thymine, cytosine, adenine, then you have the cytosine here. Okay, cytosine and guanine, okay, and one strand. Then the corresponding region, the complementary strand, will have the base sequence. As you know, it will be adenine. Then you have guanine, thymine, guanine, and cytosine, right? That is how, because of the base pairing, that is why it is complementary. Now, the DNA consists of two complementary chains. They will be twisted around each other, forming a right-handed helix. One turn of the helix measures one turn. Here it is. Yeah, one turn of the helix. It measures three. Okay, one turn is 34 nm Armstrong. Okay, and it contains 10 pairs of nucleotide. Within it, it will have 10 pairs of nucleotide placed at a regular interval of 3.4. That is the stacking between these two. Okay, that will be around 3.5. That is, whole one helix is 34, and the stacking between these is 3.4, which will give 10. So 10 base pairs per helix will be present. Okay, now the diameter of the helix um, will, as you know, it is 20 Armstrong. It's written the diameter, it is the diameter. Okay, this is, okay, you can just bring it here. This is the diameter, it is 20 nm. Okay, and there is two grooves, that is a major groove and a minor groove. Okay, now the narrow groove is the distance between the pairs. So if you can see here, this is the minor groove. Okay, this whole thing is the major groove. It's a major groove, yes, it's given. This is the major groove and this is the minor groove, right? So let's come back to the points which is mentioned here. So double helix comprises of two complementary double helix. Yes, the two polypeptide chains are wound clockwise around a central axis. So there will be a central axis, which is, it's not central, let me just draw it clearly. You will have a central axis like this. Okay. To form a circular staircase structure. Now, the sugar phosphate backbones are on the outside of the double helix, and purine and pyrimidine forms the rugs of the DNA helix ladder. Now, the phosphate groups gives the molecules a negative charge on the surface. Two DNA strands are anti-parallel. The direction of each strand is identified by the number of carbon in the sugar molecule, which I have mentioned in my previous videos. Then the 5-N is one where the carbon is not bound to the other nucleotide, and 3-N is the one where the carbon is not bound to the other nucleotide. So we have mentioned about this in our previous lecture on molecular structure of DNA. So continuing with the important features of Watson and Crick, the nitrogenous bases lie perpendicular to the long axis of the molecule, and these are stacked one on top of other like a pile of saucers. Now, the hydrophobic interaction between the stack provides this is one of the main interaction which I have mentioned earlier, that it provides a stability to the DNA. And the distance of a phosphorus atom of the backbone to the center of the axis is 10. And the width and the diameter of the helix is 20A, or the 2nm. 10 base pair per turn of the helix. The spacing, as I told you, 34 by 3.4, you get 10. So therefore, each turn of helix is 34 Armstrong, or the 3.4 nm. Okay, let's continue. As a result of double helical nature of DNA, the molecule has two asymmetrical grooves. As I mentioned, they are the larger groove and the minor groove. Larger major groove occurs when the backbones are far apart, while the smaller one is called the minor groove, which occurs when they are close together. Since the major and the minor grooves expose the edges of the bases, the grooves can be used to tell the base sequence of specific DNA. So this is all about the important features of Watson and Crick. I hope you really know now the structure of the DNA, about the history, structure. We are seeing the chemical composition as a whole. We have covered the DNA. Keep watching to know further details about DNA structure. Thank you.